Click the bell icon and subscribe to our channel so that you will not miss any classes.
Now, let us discuss production of insect resistant plants. Insect resistant plants will help us to reduce the reliance on chemical pesticides. We have already discussed what is the disadvantage of chemical pesticides. So those disadvantages can be overcome with the production of insect resistant plants. A bacteria called Bacillus thuringiensis, in short we can refer it as BT, is having a set of genes called cry genes. So when you are referring genes, you should write C small. And these cry genes able to produce some protein crystals, we can call them as crystalline protein. So in short we can refer it as CRY proteins. So here when you are referring protein, you should use C capital. And these crystalline proteins have a toxin in the inactive form that is called protoxin. Anywhere if you come across with the term pro, that is refers to inactive form. For example, proinsulin, protoxin, that is toxin in the inactive form. Proinsulin, insulin in the inactive form. Likewise, this protoxin is present in the crystalline protein that is in the inactive form. When an insect eat upon these crystalline protein, in the insect gut there is a alkaline pH. In the alkaline pH of the insect gut, the protein crystals get dissolved. Protein crystals get dissolved in the alkaline pH and the inactive protoxin is activated. So, we can tell that when an insect eat upon the crystalline proteins in which the protoxin is present, this protoxin will be activated in the alkaline pH of the insect gut where the crystalline proteins are dissolved. So as a result, the inactive toxin is get activated. When it get activated, it will binds to the gut epithelial cells. It will binds to the gut epithelial cells of the insect and it will cause us the cell swelling and lysis. Due to these two reasons, you have to remember the reasons for the death of insect. Insect will die due to the epithelial cells will get swelled and undergo lysis when the toxin binds to the epithelial cell. As a result, the insect will die. This is a natural phenomenon which is taking place. Now, biotechnologists have exploited this mechanism in order to produce the insect resistant plants. Here, the one example where I will be taking is the production of BT cotton. Here we have discussed the cry genes produces crystalline protein. And one more thing you should know here is the cry genes are of different types. For example, cry1 AC, cry2 AB, cry1 AB. So these are the different types of which are important I am referring. many are there. Now, these types of crystalline proteins will target different types of insects. The type of insects which are killed by the crigenes in general include lepidopterans, then coleopterans and dipterids. So these type of insects can be killed in general by crigenes. And I said there are different types of crigenes and they target different hosts. That is, they have the host specificity. For example, Cry1AC and Cry2AB can kill the caterpillar larva of the cotton bollworm. So I'll write in different color. It can kill cotton bollworm. So that means there is Cry1AB, Cry1AC, Cry2AB. All of these are. But all of these are not able to kill the cotton bollworms. Cotton bollworms can be killed by Cry1AC and Cry2AB. Likewise, Cry1AB can kill corn borer. So by studying these host specificities, what scientists did is they isolated Cry1AC and Cry2AB genes from Bacillus thuringiensis. That is, I will write in a flow chart. Cry1AC and Cry2AB genes from Bacillus thuringiensis were isolated and they are incorporated into cotton plant using TI plasmid of Agrobacterium tumefacium that we have studied in the previous chapter. The vectors for gene transfer into plant cell. Generally it is TI plasmid of Agrobacterium. Then Agrobacterium can clone Agrobacterium vector can clone the Cry1AC and Cry2AB genes into the cotton plant and we call it as now the BT cotton. In all parts of the plants there is production of protoxin. That is, in leaf, bud, stem, everywhere the protoxin is produced. That is, crystalline form of protein is produced. When insect eat upon insect caterpillar, that is, usually the insects will eat upon plants whenever they are present at the caterpillar stage. That are various stages of insect life cycle. That is moth, egg, caterpillar. In caterpillar is the vigorous eater. It will eat upon the plant and it will grow. So when insect caterpillar eat upon this BT cotton plant part, then what happens? We have studied already. In the alkaline pH, the crystalline proteins get dissolved. As a result, the inactive toxin is get activated. It binds to the epithelial cells of the insect gut. It causes cell swelling and lysis. As a result, the insect will die. So for this to action, it may take nearly 2 to 3 days. Within 2 to 3 days, the caterpillar will be died. So that the life cycle is stopped at one stage. So the plant crop is protected. So now this is one of the technology, that is production of insect resistant plants is one of the technology to produce more product from the plants without using any chemical fertilizers, insecticides or the pesticides. Sorry, not fertilizers, without the use of insecticides. Right? Not only cotton, there are other plant crops which are genetically modified to improve the insect resistance. So that is BT genes. Crigenes have been introduced into other plants also. Like for example, rice, soya bean, then cotton, brinjal, So likewise, in many plants, they have induced insect resistance by transferring the genes of Bacillus thuringiensis. That's all about the production of insect resistant plants. And we shall take up the next class, the production of pest resistant plants.